Chemiosmotic hypothesis. During photosynthesis, there is release of two energy-rich compounds ATP and NADPH. Chemiosmotic hypothesis explain how ATP is produced in chloroplast during photosynthesis. As we know, each chloroplast consists of a membranous system called grana. Grana consists of thylakoid. Thylakoid is made up of a membrane and the lumen. There is a accumulation of proton within the lumen which results in a development of proton gradient across the thylakoid membrane. That is, in stroma, decreased concentration of H+, in lumen, increased concentration of H+. For this, there are three reasons. First reason is splitting of water occurs inside the membrane of the thylakoid. That is, splitting of water is associated with photosystem 2 and this is present towards the lumen of thylakoid. The product of splitting of water is H+ ions and half molecule of oxygen. Oxygen is evaporated as a gas. H+ ions are added to the lumen which increases the concentration of H+. As a result, there is release of electrons also. These electrons are given to photosystem 2. Later, these electrons from photosystem 2 are given to a primary electron acceptor. The second reason is the primary electron acceptor will receive the electron as well. It also receives the H+ ions from the stroma because later it will donate the electron to a proton carrier, not to an electron carrier, that is cytochrome B6F. During the transport of electrons through this, there is a pumping of H+ ions from stroma into the lumen of thylakoid. As a result, within the lumen, there is an increased concentration of H+ and decreased concentration of H+ in the stroma. The third reason is the NADP reductase enzyme is associated with photosystem 2 is present towards the stroma. The function of NADP reductase enzyme is to reduce NADP to NADPH. For this, there is a need of H+ and those H+ are from stroma. Now, H+ ions are removed from stroma during this process. Now, because of these three reasons, there is a increased concentration of H+ in lumen and decreased concentration of H+ in stroma. As a result, we know that all the systems will try to be in an equilibrium. So, to maintain equilibrium, from lumen, there is a pumping of H+ ions towards the stroma through F0, F1 particle, which releases energy, and that energy is utilized for the conversion of ADP to ATP. This is how ATP is synthesized.